Welcome to the deep dive. Today, we're embarking on really an incredible journey into the most complex machine known, your nervous system.
Absolutely. It's just mind-boggling stuff.
We're going to peel back the layers, starting right down at the fundamental building blocks, and see how these tiny parts, well, how they build up this intricate network that lets you think, feel, experience everything.
Yeah, our mission today is basically to navigate from, the microscopic world of a single nerve cell and neuron
Hmm.
all the way up to the grand structures, the brain, the spinal cord,
Mm-hmm.
and we'll uncover the specialized designs, how they enable everything from just a quick thought to a complex movement. It's not just what's there, but why it's built that way, why it matters.
Exactly. Get ready for some, I think, genuine aha moments. It might change how you think about your own body.
Definitely.
So we'll build this understanding piece by piece, seeing how each bit contributes to the whole symphony.
Sounds good.
Okay, so let's zoom right in. The star of the show, the neuron.
Right, the nerve cell.
At its core, you've got the cell body, a kind of bustling control center. What are the absolute essentials happening inside? What makes it such a powerhouse?
We've got it, the cell body, or perikaryon. Crucially, it houses the nucleus, the cell's command center with all the DNA.
Yeah.
And it's packed with mitochondria, little power plants, churning out energy. Neurons need a lot of energy.
Oh, completely on.
Exactly. But something really distinctive and vital for communication is this stuff called Nissl substance.
Ah, Nissl substance.
Yeah, you can see it clearly with certain stains. It's basically rough endoplasmic reticulum, loads of it. And what that tells us is there's a phenomenal level of protein synthesis going on.
So it's constantly building things.
Constantly. It's like a miniature factory, always making proteins for maintenance, repair, producing neurotransmitters, enzymes, everything needed to send and receive signals, thousands per second.
Wow.
And this relentless activity, it underlines why they're so metabolically demanding, why they need that constant energy supply. They're vulnerable if it dips.
That's a great point about the energy needs. Does that high activity also mean other stuff accumulates, like those pigment granules you sometimes see?
Absolutely. Good question. For instance, certain neurons, like in the substantia nigra important for movement, they accumulate neuromelanin.
Okay.
And, as neurons age, you often see a buildup of lipofuscin. It's like wear and tear markers. They give clues about the neuron's life or its specific job.
Interesting. Little biological footprints. Okay, so we have this busy cell body, but how does it actually connect? How does it talk across distances? Sometimes huge distances in the body. That's where the extensions come in, right? The neurites?
Precisely. Neurons are all about connection.
Yeah.
They reach out with two main types of these processes, or neurites,
Mm-hmm.
dendrites and axons.
Right?
Dendrites are usually shorter, highly branched. Think of like a tree's branches spreading out, often covered in tiny bumps called spines.
Spines.
Yeah, dendritic spines that massively increase the surface area. And dendrites are the main receiving antennas they draw signals in towards the cell body.
Okay, so dendrites are pulling information in. Why so branched? Why the spines? Why not just a simple input wire? What's the advantage?
Great question. That branching and the spines, it exponentially increases the surface area. It means one neuron can receive input from thousands, even tens of thousands of other neurons simultaneously.
Oh, thousands.
Oh, yeah.
Yeah.
It's all about integration. Like listening to a whole crowd of conversations at once, weighing all that incoming data. It's an incredible design for complex information gathering.
Okay, that makes sense. So integration station. And the other extension, the axon.
Right. The axon, that's usually a single, much longer and thinner process. It stretches away from the cell body. Unlike dendrites, it tends to have a pretty uniform diameter, smoother surface.
Okay.
The axon's job, the outgoing transmission cable, it carries the nerve impulse, the signal, away from the cell body, sometimes, all the way down your leg.
Incredible length.
Absolutely.
Yeah.
Now, axons don't usually branch much near the cell body, but they can send off side branches called axon collaterals. And then at the very end, they split into many fine processes called axon terminals. That's where the signal actually gets passed on.
Got it. So the axon is the long distance line, and these axons bundled together, that's what we mean by it, a nerve fiber, right?
Yeah.
And then bundles of those make up nerve tracts in the brain or peripheral nerves.
Exactly. You've got it. Axons are the nerve fibers, bundles in the CNS or tracts, bundles in the PNS or peripheral nerves. That's the wiring.
Okay. And where does the signal, the electrical impulse, actually start its journey down the axon? Is there a specific launch point?
Yes, absolutely critical point. It's called the axon hillock in the initial segment. This is a little cone-shaped area where the axon leaves the cell body and the very first part of the axon itself.
Okay.
The zone is special. It doesn't have that Nissl substance we talked about, but it's packed, absolutely packed, with voltage-sensitive ion channels.
Ah, the channels that open and close for the electrical signal.
Precisely. This is effectively the neuron's trigger zone, or decision center.
Mm.
All the signals coming in from the dendrites get summed up here. If the total input reaches a certain threshold, bang, an action potential. The electrical nerve impulse is generated. It fires.
Boom. Decision made. Signal sent.
That's it. And it then travels or propagates all the way down the axon.
It's like the trigger, the firing pin.
And I've heard the inside of the neuron isn't static either. There's constant movement.
That's right. The cytoplasm inside, especially down the long axon, is in constant motion. It's called exoplasmic flow or axonal transport.
Exoplasmic flow. Okay.
It's a really sophisticated transport system. There's a slow component and a fast one. It moves vital materials, proteins, enzymes, bits needed for neurotransmitters away from the cell body, down the axon. And it also brings back waste products or signaling molecules towards the cell body.
Like a two-way street?
Exactly. It's the
Yeah.
essential supply chain, especially for those really long axons. Got to keep the distant terminals supplied and get feedback. Makes sense. Okay, so we have the signal, the action potential, firing down the axon. But speed is often crucial, right? How does the nervous system ensure messages get there fast?
Now we get to something really ingenious. Myelin.
The myelin sheath. I've heard of this. It's like insulation.
It is, but it's much more than just passive insulation. It's a high-performance accelerant. It's a fatty layer wrapped around many, though not all, axons.
Made of fat.
Mostly, yeah. It's a complex mix of proteins, lipids like cholesterol and phospholipids, glycosphingolipids, and water. This composition gives it excellent electrical insulating properties.
And that speeds things up.
Dramatically. Without myelin, our nervous system would be incredibly slow. Reaction times would be sluggish, complex thought, probably impossible.
Wow. And it's not just there, it's actively built, right, by specific cells.
That's right. In the peripheral nervous system, the nerves in your limbs and body, Schwann cells, do the job.
Schwann cells. Okay.
Picture this. An axon kind of sinks into a Schwann cell. The Schwann cell then wraps its own cell membrane around and around and around the axon, layer upon layer, like rolling up a sleeping bag really tightly.
Okay, I can picture that.
And lipids get packed in between these membrane layers, forming the thick, insulating myelin sheath. Importantly, a thin layer of the Schwann cell cytoplasm stays on the outside, called the neurilemma. This is vital for potential nerve repair if there's damage in the PNS.
Neurilemma, got it. Crucial for repair. And in the brain and spinal cord, the CNS.
Different cell. In the CNS, it's the oligodendrocytes. They provide a similar myelin covering wrapping around axons there.
Oligodendrocytes. So Schwann cells outside, oligodendrocytes inside.
You got it.
Now, I remember reading that this myelin sheath isn't continuous. There are gaps.
Exactly. Crucial point. There are these short, unmyelinated gaps at regular intervals along the axon. They're called the nodes of Ranvier. The myelinated segment between two nodes is called an internode.
Nodes of Ranvier. Okay, gaps in the insulation. Why? That seems counterintuitive if you want speed.
But that's the genius of it. These nodes are where the action happens, electrically speaking. The voltage-gated ion channels needed for the action potential are concentrated at the nodes.
Okay. Oh.
So instead of the electrical signal having to regenerate continuously along the entire axon membrane, which happens in unmyelinated axons and myelinated axons, the signal effectively jumps from one node of Ranvier to the next.
Jumps, like leapfrog.
Exactly like leapfrog. This process is called saltatory conduction. Saltare is Latin for to leap.
Saltatory conduction. Okay, that sounds fast.
Incredibly fast. It boosts the speed of nerve impulse transmission hugely, maybe up to 100 times faster than an unmyelinated axon of the same size.
Wow. So the nodes are like little relay stations, allowing the signal to skip the insulated parts?
Precisely. Think about touching something hot. You need that signal to get to your brain and back to your muscles almost instantly. Saltatory conduction, thanks to myelin and the nodes of Ranvier, makes that possible. It's a critical adaptation.
Amazing. Myelin, the speed booster.
But are all neurons built the same way, or are there different types for different jobs?
Oh, definitely different types. Neurons are incredibly specialized. We can classify them in a few key ways. One is by the number of processes, or neurites, extending from the cell body.
Okay.
You have unipolar neurons. They have just a single process that comes off the cell body and then splits into two branches, one acting like a dendrite, one like an axon. You find these mainly in sensory ganglia, like the ones bringing touch information from your skin.
Unipolar. Got it. One pole splitting.
Then there are bipolar neurons. These have one axon extending from one end of the cell body and one dendrite from the other end. Pretty straightforward. You find these in specialized sensory systems like the retina of your eye or in your inner ear for hearing and balance.
Bipolar. Two distinct poles. Makes sense.
And then the most common type by far, multipolar neurons. These have one axon but many dendrites branching out from the cell body. Most neurons in your brain and spinal cord, including motor neurons controlling muscles, are multipolar. Multipolar, okay, the most common. One axon, lots of dendrites. So structure varies. What about function?
Functionally, we make a broad distinction too. You have sensory neurons, also called afferent neurons. They carry nerve impulses from sensory receptors in your skin, eyes, ears, internal organs towards the central nervous system. They're the reporters.
Sensory in, afferent towards. Okay.
And then you have motor neurons, or efferent neurons. They transmit impulses away from the CNS to effector organs, mainly muscles and glands. They carry the commands efferent for exit.
Motor out, efferent away. Got it. Sensory reporters, motor commanders.
Exactly. And there's even another classification based on axon length. Golgi-type I neurons have really long axons that connect distant regions, like the big pyramidal cells going from your brain's motor cortex down the spinal cord.
Long-distance callers.
Yep. And Golgi-type II neurons have short axons that often terminate very close to their own cell body. These are typically interneurons, involved in local processing within a brain region, like in the cerebral or cerebellar cortex.
Local circuit specialists. Okay, so a huge diversity in shape, function, and reach.
Incredible diversity, all tailored for specific roles in the network.
All right, we've dug into the neuron, the star communicator, but you mentioned earlier they don't work alone. They have this massive support crew, the neuroglia.
Ah, yes, the glial cells. The unsung heroes, truly.
Unsung, but numerous, right? You said way more numerous than neurons.
Astonishingly so.
Yeah.
Estimates vary, but yeah, easily 10 to 50 times more glial cells than neurons in your brain and spinal cord. They're much smaller, but they vastly outnumber the neurons.
10 to 50 times. Wow. So what are all these support cells actually doing? They're not just packing material, are they?
Oh, absolutely not. They're incredibly active and perform vital roles. They provide structural support, sure, but also metabolic support. They form myelin, they clean up debris, they help regulate the chemical environment around neurons, and some even seem to play a role in signaling themselves.
Okay, let's break that down. What are the key types? You mentioned oligodendrocytes already for CNS myelin.
Right. And Schwann cells for PNS myelin. But maybe the most abundant type in the CNS are the astrocytes.
Astrocyte star cell, right.
Exactly. They're star-shaped with lots of radiating processes. Think of them as the brain's multitasking caretakers. They provide mechanical support, kind of hold things together. They act as insulators between neurons. Critically, they help maintain the right chemical environment. They mop up excess potassium ions, and they take up neurotransmitters like glutamate or GABA after they've done their job at the synapse, helping to turn signals off.
Okay, so regulating the local environment. That sounds important.
Hugely important.
Yeah.
And maybe their most famous role, they contribute significantly to forming the blood-brain barrier. Their little end feet wrap around capillaries, forming a tight seal that controls what gets from the blood into the brain tissue, protects the brain.
The gatekeepers.
Precisely. They also help repair damaged areas by forming glial scars, a process called gliosis. And fascinatingly, astrocytes communicate with each other through gap junctions and using calcium signals. They seem to actively listen in on and modulate synaptic activity.
Wow, so they're not just passive support at all. They're actively involved in brain function.
Absolutely. And there are different types: fibrous astrocytes, mainly in white matter; protoplasmic astrocytes, mainly in gray matter, each adapted slightly.
Okay, astrocytes, oligodendrocytes, what else?
We have the ependymal cells. These are the cells that line the fluid-filled cavities within the brain, the ventricles, and the central canal of the spinal cord.
Lining the internal spaces.
Right. Some specialized ependymal cells form the choroid plexus, which is where cerebrospinal fluid, CSF, is produced. So they're involved in making CSF and helping it circulate, and also in exchanging substances between the brain tissue and the CSF.
CSF production and interface. Got it. And you mentioned cleanup crew.
Ah, yes, the microglia. These are the smallest glial cells. And uniquely, they actually originate from a different embryonic layer, the mesoderm. They're related to immune cells like macrophages.
So they're the brain's resident immune cells.
Pretty much. They're normally in a resting state, kind of surveying their territory. But if there's injury, infection, or inflammation, they activate. They transform into phagocytes, big eaters, and engulf dead cells, debris, pathogens. They're the essential cleanup and defense crew of the CNS.
Microglia, the defenders and janitors. Okay. So astrocytes, oligodendrocytes, ependymal cells, microglia, that covers the main CNS scalia.
Those are the big four in the CNS, yes. Plus, we already mentioned Schwann cells in the PNS for myelin. And surrounding the neuron cell bodies in peripheral ganglia, you have capsular cells or satellite cells, which are kind of analogous to astrocytes, providing support and regulating the environment in those peripheral nerve clusters.
Right. Satellite cells in ganglia. Okay, what a team. Neurons doing the communicating, glia doing, well, almost everything else, it seems.
It's a true partnership. You can't understand nervous system function without appreciating the neurolia.
Absolutely. Okay, so we have the cells, neurons, and glia. We know neurons have axons to send signals. How do they actually pass that signal from one neuron to the next? This is the synapse, right? The connection point.
Exactly. The synapse is the crucial junction where information flows between neurons. It's where the magic happens, really.
And there are different kinds.
Broadly, two main types.
Yeah.
Electrical synapses and chemical synapses.
Electrical sounds. Well, electric. Fast.
Very fast. In an electrical synapse, the membranes of the two neurons are extremely close, only about 4 nanometers apart.
Sure.
And they're directly connected by channels formed by proteins called connexins.
Connexins, like little tunnels.
Exactly. These form gap junctions, allowing ions the electrical current to flow directly from one cell into the next. It's almost instantaneous transmission.
Yeah.
Very good for synchronizing groups of neurons, like for rhythmic activities or rapid reflexes.
Direct connection, super fast sync.
Yeah.
Okay. But I gather chemical synapses are more common.
Much more common, especially in the mammalian brain. They offer more flexibility and potential for modification, which is key for learning and memory.
Ah, okay. So how does a chemical synapse work? It involves chemicals, neurotransmitters.
Precisely. A chemical synapse has 3 main parts. You have the end of the axon from the sending neuron, the presynaptic terminal, often called a synaptic bouton or bag.
Presynaptic before the gap.
Right. Then there's the membrane of the receiving neuron, the postsynaptic membrane, which could be on a dendrite, or the cell body.
Postsynaptic after the gap.
Yep. And between them is a physical gap, typically 20-40 nanometers wide, called the synaptic cleft.
The gap itself. Okay.
So the electrical signal, the action potential, arrives to the presynaptic terminal. This triggers the release of chemical messengers, neurotransmitters stored in little sacs called vesicles. These neurotransmitters diffuse across the synaptic cleft.
Float across the gap.
And bind to specific receptor proteins embedded in the postsynaptic membrane. This binding then causes a change in the postsynaptic neuron, either exciting it or inhibiting it, making it more or less likely to fire its own action potential.
So the electrical signal gets converted to chemical, crosses the gap, and then potentially converts back to electrical in the next neuron.
That's the essence of it. And it's not just a simple relay. This is where information processing happens. The type of neurotransmitter, the type of receptor, how many receptors are activated, all this influences the outcome.
And you mentioned modification. This is where learning happens.
Fundamentally, yes. The strength or efficiency of these chemical synapses can change with experience. They can become stronger or weaker. This synaptic plasticity is thought to be the cellular basis of learning and memory. It's incredibly dynamic.
Wow. And these synapses can form between different parts of neurons, right? Not just axon to dendrite.
Oh, yes. The most common is axodendritic, axon to dendrite. But you also have axosomatic, axon to cell body, axoraxonal, axon onto another axon terminal, often for modulation, even dendrite to dendrite sometimes. Lots of connection possibilities.
It's an incredibly intricate communication network at the finest level.
Absolutely mind-boggling complexity, all happening at these tiny junctions.
Okay, so we've gone from the neuron, its extensions, its support crew, the myelin, the synapse. Now, how do these individual nerve fibers, these axons, get bundled together to form the actual nerves we can see, the cables running through our body?
Right, taking a step back up in scale. Individual nerve fibers, the myelinated or unmyelinated axons with their Schwann cells in the PNS, don't just float around. They're organized and protected by layers of connective tissue.
Like insulation and casing around electrical wires?
Very similar analogy. Around each individual nerve fiber, there's a delicate layer of connective tissue called the endonerium.
Endonerium, innermost layer.
Then groups of these fibers are bundled together into what's called a fasciculus or fascicle. Each fascicle is wrapped by a thicker, stronger layer called the perineurium. This layer is important. It forms a diffusion barrier, helping maintain the nerve's internal environment.
Perineurium around a bundle, a fascicle. Okay.
And finally, the entire nerve trunk, which might contain many fascicles bundled together, is enclosed by the outermost dense connective tissue sheath, the epineurium. This outer layer often contains fatty tissue, which cushions the nerve fibers.
Epineurium, the tough outer coat. So endo, peri, epi, layers of protection.
Exactly. And it's clinically relevant, too. You mentioned fat cushioning. If someone is severely malnourished or bedridden for a long time, they can lose this fat. Nerves might get compressed against bone, leading to pressure palsies.
Wow, that highlights the importance of even the fatty tissue. And blood supply runs through these layers, too.
Yes, the blood vessels supplying the nerve, the vasa nervorum, travel within these connective tissue sheaths. If that blood supply gets compromised, say by compression or vascular disease, you can get ischemic neuritis nerve damage due to lack of oxygen, which can be very painful.
So the structure is vital for protection and blood supply. Makes perfect sense.
Now, what about those little clusters of nerve cell bodies outside the brain and spinal cord? The ganglia.
Right, ganglia. These are essentially collections of neuron cell bodies located outside the CNS. Think of them as relay stations or peripheral processing centers.
And there are different types here, too.
Primarily two main types based on function and structure: sensory ganglia and autonomic ganglia.
Okay, sensory versus autonomic. What's the difference?
Sensory ganglia, like the dorsal root ganglia associated with spinal nerves or ganglia on some cranial nerves, mostly contain the cell bodies of sensory neurons, those unipolar ones we talked about.
Bringing sensory info in? Exactly. Structurally, the neurons in sensory ganglia are typically large, often arranged in groups near the periphery of the ganglion. And each neuron cell body is surrounded by a layer of supportive capsular cells or satellite cells. They also tend to have lots of myelinated nerve fibers running through them.
Organized large cells, myelinated fibers,
Okay.
and autonomic ganglia.
Autonomic ganglia are part of the autonomic nervous system, controlling involuntary functions like heart rate, digestion, gland secretion. The neurons here are typically smaller and multipolar. They're usually scattered more randomly throughout the ganglion, not in neat groups.
Smaller, scattered, multipolar.
Right. And their nerve fibers are often thinner and non-myelinated. While they do have satellite cells around the neuron bodies, they're often less prominent or well-defined than in sensory ganglia. Also, the nissl substance inside these autonomic neurons can be quite prominent, reflecting their constant activity.
Different structure reflecting their different job in controlling bodily functions.
Form follows function again.
Okay, fascinating. Now, let's zoom out even further to the big structures, the brain and spinal cord, the central nervous system. How is the tissue organized there on a large scale?
At the macroscopic level, the most obvious distinction within the CNS is between gray matter and white matter.
Gray and white, what defines them?
Gray matter consists mainly of neuron cell bodies, dendrites, unmyelinated axons, and glial cells, particularly astrocytes and microglia. This is where most of the synaptic communication and information processing actually happens, the computing centers.
Okay, gray matter is the processing hubs, and white matter.
White matter is composed primarily of myelinated axons. The myelin sheaths, being fatty, give this tissue its whitish appearance. White matter consists of the tracks, the bundles of axons transmitting signals over longer distances between different gray matter regions or between the brain and spinal cord. It's the wiring, the communication highways.
Got it. Gray matter processes, white matter transmits. How are they arranged?
The arrangement actually differs. In the spinal cord and the brainstem, generally, the white matter's on the outside, forming large columns or tracks, while the gray matter is located more centrally, often in an H shape or butterfly shape in the spinal cord cross-section.
White outside, gray inside for spinal cord brainstem.
Right. But in the two largest parts of the brain, the cerebrum and the cerebellum, this pattern is reversed. There, the gray matter forms a complex folded outer layer called the cortex, the bark of the brain. And the white matter lies underneath this cortex, connecting different cortical areas and linking the cortex to deeper structures.
Gray matter cortex on the outside, white matter deep inside for the cerebrum cerebellum. Interesting reversal.
It is. And within the deeper parts of the cerebrum and cerebellum and in the brainstem, you also find distinct clusters of gray matter embedded within the white matter. These isolated clumps of neuron cell bodies are called nuclei.
Nuclei, like little gray matter islands in the white matter sea.
Good way to put it. And the bundles of white matter axons connecting these nuclei or different cortical regions are called tracks, or sometimes fasciculi, ventriculi, lemnisci, peduncles. Various names for bundles of fibers.
Okay. Let's look quickly at the spinal cord structure then, that H-shaped gray matter.
Yes, in a cross-section of the spinal cord, the central gray matter looks like an H. The front projections are the anterior or ventral gray horns, containing mostly motor neuron cell bodies. The back projections are the posterior or dorsal gray horns, receiving sensory information. In some parts of the cord, there's also a lateral gray horn related to the autonomic nervous system.
Motor in front, sensory in back.
Generally, yes. And connecting the two sides of the H is the gray commissure, with the tiny central canal running through it, which contains CSF and is lined by those ependymal cells. The surrounding white matter is divided into posterior, lateral, and anterior funiculi columns, containing the ascending, sensory, and descending motor tracts.
The information highway clearly organized. Now, the cerebellum, the little brain, crucial for coordination, balance, has a really distinct structure, right? Very uniform.
Extremely distinct and remarkably uniform across its entire cortex. The cerebellar cortex has a beautiful, precise three-layered structure.
Layers.
On the outside in. First is the molecular layer. It's relatively cell-sparse, containing mostly axons and dendrites, but also some inhibitory interneurons called stellate cells and basket cells. Basket cells are interesting. Their axons form a basket around the cell bodies of the next layer down,
Which is
the Purkinje cell layer. This is the most characteristic layer. It's just a single thin layer composed of huge flask-shaped neurons called Purkinje cells.
the famous Purkinje cell.
Indeed. They are magnificent cells. Each one has an incredibly elaborate flattened dendritic tree that extends up into the molecular layer like a FAM or an espaliered tree. It receives a massive amount of input.
So they're integrating tons of information.
Absolutely.
Yeah.
And what's really key is that the axon of the Purkinje cell is the sole output of the entire cerebellar cortex. All the complex processing within the cortex converges onto these Purkinje cells, and they send the final signal out.
The only way out is through the Purkinje cell axon.
Wow. And what kind of signal do they send?
Inhibitory. They use the neurotransmitter GABA. So the main output of the cerebellar cortex is inhibitory, projecting to the deep cerebellar nuclei located in the white matter underneath the cortex. This inhibition is crucial for refining movement, coordinating timing, and motor learning.
So the cerebellum fine-tunes movement largely by saying stop or less.
In essence, yes. It sculpts motor commands through precisely timed inhibition. Below the Purkinje layer is the third layer, the granular layer.
Granular layer.
Sounds packed.
It is incredibly densely packed with tiny neurons called granule cells, possibly the most numerous neuron type in the entire brain. There are also some larger inhibitory interneurons called Golgi cells here.
Okay, molecular, Purkinje, granular. How does information flow in?
Information comes into the cerebellar cortex via two main types of excitatory afferent fibers. Mossy fibers originate from many different areas in the brainstem and spinal cord, carrying diverse sensory and motor context information. They synapse primarily on the granule cells in complex structures called glomeruli.
Mossy fibers to granule cells? Okay.
The granule cells then send their axons up into the molecular layer, where they split to form parallel fibers that run horizontally, making contact with the dendrites of many Purkinje cells.
So mossy fibers provide broad context via granule cells. What's the other input?
The other input is via climbing fibers. These originate exclusively from a nucleus in the brainstem called the inferior olive. Each climbing fiber makes a very strong, powerful synapse directly onto the dendritic tree of one Purkinje cell. It climbs up the dendrites.
One climbing fiber to one Purkinje cell. Very specific.
Extremely specific and powerful excitatory input. It's thought to convey error signals or teaching signals, crucial for motor learning. So you have this interplay. Broad context from mossy fibers via granule cells and specific error signals from climbing fibers, all converging onto the Purkinje cells, which then compute the appropriate inhibitory output.
It's an incredibly elegant computational circuit.
It really is. A beautiful example of structure-enabling function.
Okay, finally, the grand finale. The cerebral cortex, the seat of consciousness, language, reason, the most complex part.
Indeed. The cerebral cortex is where our highest mental functions reside. Structurally, it's also layered, typically described as having 6 distinct layers, or laminae, although the thickness and cell density of these layers vary across different cortical areas.
6 layers, more complex than the cerebellum's 3. What are the main cell types?
The principal neurons are the pyramidal cells. They get their name from their triangular or pyramid-shaped cell body. They typically have a large apical dendrite extending upwards towards the cortical surface and several basal dendrites spreading out horizontally. Their axons usually travel downwards, either to other cortical areas or out of the cortex entirely. They make up maybe 2/3 of cortical neurons and are generally excitatory using glutamate.
pyramidal cells the main output and association neurons.
Right. The other major class are stellate neurons, or star-shaped cells. These are usually smaller, multipolar neurons. Many are interneurons, meaning their axons stay within the local cortical area involved in processing and modulating activity. Some are excitatory, some inhibitory, using GABA. They make up about a third of cortical neurons.
Pyramidal and stellate. Okay, the 6 layers, what distinguishes them?
They're numbered I to 6th, from the outermost surface down to the underlying white matter. Layer I, molecular layer, mostly axons and dendrites, relatively few cell bodies. Layer 2, external granular layer, densely packed with small stellate cells and some small pyramidal cells. Layer 3, external pyramidal layer, contains medium-sized pyramidal cells, major source of cortical connections. Layer 4, internal granular layer, main recipient layer for sensory information coming from the thalamus, rich in stellate cells. Layer V, internal pyramidal ganglionic layer, contains the largest pyramidal cells, especially in motor cortex, including the giant bed cells. This is the major output layer to subcortical structures, like the brainstem and spinal cord. Layer 6, multiform layer, a more diverse layer with various cell types blending into the white matter below, projects mainly to the thallus.
Wow, okay, so different layers have different inputs, outputs, and cell populations. It's highly organized.
Very much so. Inputs tend to arrive in layer 4. Processing occurs within and between layers, especially 10 and 3, and outputs leave primarily from layers V and 6.
And you mentioned variations. The layers aren't the same everywhere.
Correct. The relative thickness and prominence of these layers vary depending on the function of that cortical area. For example, primary motor cortex, area 4, which sends commands down to the spinal cord, has a very thick layer V, packed with huge pyramidal cells, and relatively thin granular layers, second 4. We call this a granular cortex.
A granular, less granular, more output-focused. Makes sense for motor areas.
Exactly. Conversely, primary sensory areas, like the visual cortex, area 17, which receive massive input from the thalamus, have very prominent granular layers, especially layer 4, and less developed pyramidal layers, 3 and V. We call this granular cortex, or coniocortex.
Granular, well-developed granular layers for receiving sensory input.
Right. In fact, layer 4 in the primary visual cortex is so thick and packed with myelinated fibers receiving input that it's actually visible to the naked eye as a white stripe, the stria of Gennari. That's why it's also called the striate cortex.
The stria of Gennari. Amazing detail. So what this all really means is that the cortex isn't uniform. Its fine structure, its lamination, is tailored specifically for the job that region does, whether it's seeing, hearing, moving, or thinking.
Perfectly put. It's an incredible example of specialization, how evolution has shaped the cellular architecture to optimize function, right up to the highest levels of cognition.
What an incredible journey we've taken today. From, the single busy neuron and its extensions, the crucial role of myelin, the vital support of the glial cells,
Through the intricate dance at the synapse, the organization of nerves and ganglia.
all the way up to the layered complexity of the spinal cord, the cerebellum, and the cerebral cortex. It's just a masterpiece of biological engineering working inside us right now.
It really is breathtaking when you break it down. The sheer elegance of how it's all put together, specialized cells, precise connections, layered structures, creating this system with such immense capability. Every detail, from Nissl substance to myelination patterns to cortical layers, it all contributes.
Absolutely. And thinking about all this, especially the often overlooked support cells, it leads to a really interesting final thought for you, our listener, to ponder.
Yeah, considering that neuroglial cells, the glia, are not just passive scaffolding, but are, 10 to 50 times more numerous than neurons. And they perform this huge range of critical active functions, managing the environment, forming the blood-brain barrier, cleaning up, even actively modulating synapses through things like calcium signaling.
Oh, you're really involved.
How much of what we consider intelligence or consciousness or just the overall sophisticated function of our nervous system, how much of that might actually depend on or even emerge from the complex actions and interactions of these neuroglia, not just the neurons themselves?
How much of the thinking is done by the support crew?
Exactly. We tend to focus on the neurons as the stars, but maybe the glial cells are equally important players in the cognitive orchestra. Something to definitely think about on your next deep dive.